Let's continue our discussion on selectins. Let's see a little bit more about their structure and function. Selectin also, in fact, all molecules, adhesion molecules, have to interact with the cytoskeleton. I'll show that to you why it is necessary, but for here, I just want to mention that again, it is our selectin that we have, the structure of the selectin that we have seen previously. This selectin molecules, as I have mentioned, three domains: extracellular, transmembrane, and the cytoplasmic domain. Cytoplasmic domain of the selectins interact with anchor proteins that link the cytoplasmic domain of selectins to actin filaments. We have seen that in case of cadherins, that they are also linked to actin filaments or intermediate filaments depending upon where they are present. So again, all adhesion most adhesion molecules have to be linked to cytoskeleton in order to effectively play their role. I'll show that to you again. Why is that necessary? So let's look at the importance of attachment of cytoskeleton to adhesion molecules. I am going to give you a little example that I drew. So here we have a ball which is floating in a cup or in a bowl of water. This red ball is attached to a wooden board by a string. So this string is attaching that red ball to the wooden board. But this ball is not attached to the cup in any way. So now if we apply force and move the cup will easily move. The ball will just be dangling in the water. There's no problem moving the cup. Now if we do this experiment in a different way, if we, for example, now bind this red ball with a string to the side of a cup, now if we apply the force, the cup will not move. That makes logical sense. Here I would like to mention that in this our analogy, the wooden board is basically the ECM, or the extracellular matrix, or any type of cell junction. And this bond, this rope here, basically represents the bond between the neighboring cell or extracellular matrix with the adhesion molecule. Our red ball is basically the adhesion molecule. So if the adhesion molecule is not attached to something solid, the cup will move. It will not resist that force. However, now if in the next experiment, when we attach this ball to the side of the cup, the side of this cup is solid structure. And in our example, it is representing basically the cytoskeleton. So if now we apply the force, the cup will not move because it's attached to something solid. The link between the red ball, the string between the red ball, which is attaching red ball to the side of the cup, you can think of it as the molecules that link basically your adhesion molecule with the cytoskeleton. So now I hope it is a little bit more clear why we need to attach adhesion molecules to the ECM, because just like water, the plasma membrane is fluid. Things just move around the plasma membrane in any way. With this plasma membrane will not resist movement. So here's the reason why we need to attach our adhesion molecules directly or indirectly to the cytoskeleton. Coming back to our example, at the site of inflammation, the endothelial cells which switch on expression of selectins that recognize oligosaccharides on the white blood cells, it will just basically slow down the white blood cells. The white blood cells are still moving. Now what happens is once this interaction takes place, this interaction will result in activation of another type of molecule in WBC or the white blood cell, and this molecule can bind a specific structure on the endothelial cell, on the surface of endothelial cell in a very tight manner. So our white blood cell will no longer be able to move on the surface of endothelial cells. It is bound there. It is a relatively much stronger interaction. So this interaction, we'll talk about this interaction later on. This is mediated by another type of sticky molecule, which are called integrins. This interaction results in keeping the white blood cell there for relatively longer, so it can Now, once this interaction has taken place, the white blood cell will have time to crawl through the adjacent between the two cells and go to the site of infection. For example, this was the site of infection. The endothelial cells here were expressing the selectin, which was slowing down, first of all, causing the WBC to roll and roll and roll. Ultimately, WBC activated another type of sticky molecule called integrin, which made this white blood cell stick at the particular site and allowed the white blood cell to not only cross the space between the cells, but also cross the basal lamina or the extracellular matrix through which it has to go through in order to find the infected site or the damaged site. So the moral of the story is that number one, adhesion molecules need to be attached to something solid, a solid structure, not just a flimsy plasma membrane. They need to be attached to the cytoskeleton. Number two, adhesion molecules collaborate with each other. Different types of these adhesion molecules collaborate with each other to perform a specific function for the cells.